Hi everyone, it's a great honor to give a talk at IASP. So we originally planned to give this talk last year, but due to COVID-19, I'm giving this talk 1 year later. Early this year, this work has been published in Nature Medicine, and I will walk you through the details of the study and also add more timely discussion on the topic at the end of the talk. So I have nothing to disclose. So pain is complex and subjective experience. So there are a wide range of pain duration and a large number of descriptors in order to describe the qualitative experience of pain. Therefore, we need to go beyond examining brain responses to phasic pain stimulus that usually last around 10 to 20 seconds, and also collecting pain ratings on a one-dimensional visual analog scale. But many existing neuroimaging markers for pain use fMRI activation patterns for phasic experimental pain, which might not be generalizable to chronic pain. And the connectivity patterns are likely to play a more important role in explaining and understanding chronic pain, and this has been proposed by many different research groups. And also, there is a need for connectivity-based markers that can be used to measure or predict sustained pain. And so to utilize functional connectivity for further predictive modeling, we need to induce pain for a long time. And to obtain stable connectivity estimates induced by pain, we decided to use Tonic experimental pain task with a hypothesis that it will provide an intermediate target for modeling clinical pain. So we developed a new tonic pain task using hot sauce. So in the United States, we used Tabasco habanero hot sauce, and in the South Korea, we used capsaicin extract that we can easily get from a grocery store. And given that we are using food ingredient to induce pain, we were able to convince IRB and participants of the safety relatively easily. But a small amount of hot sauce on the tongue can induce strong pain sensation in participants for around 10 minutes. And the capsaicin is known to activate the TRPV1 channel, which is abundant in our mouth. And the orofacial pain is known to be more severe and emotionally draining than body pain. So our experimental design was quite simple. So there was a capsaicin run and control run. So during capsaicin run, we asked pain intensity and unpleasantness every 45 seconds. Thus, the tonic pain task was basically a resting state design with pain ratings. For specificity testing, we also included bitter taste and aversive odor conditions. They are aversive but non-painful conditions in study 2 and 3. So here I want to show how we actually did it using the video abstract. I will play a part of the video that shows the procedure. Here is the filter paper with a small amount of capsaicin-rich hot sauce on it. So in the real experiment, we ask participants to place this filter paper on the tongue. So I will give this to Jae-jung, and participants lie down. So actually the person who is lying down there is Jae-jung, the first author of the paper. In the MRI machine, and please place the capsaicin filter paper. So we ask participants to put the filter paper in the mouth for 30 seconds, and after that we ask participants to remove the filter paper. Can you remove the filter paper? And open the mouth and breathe only through the mouth for 1 minute. This procedure will remove the liquid and settle the capsaicin on a specific area of the tongue. After that, we ask participants to close their mouth, and that will provide some heat to the tongue, and it will induce the painful sensation. We invented this procedure to keep participants from coughing in the MRI machine, and this procedure was simple and effective. So the first research question was, can we identify a brain marker of tonic pain intensity based on the whole brain functional connectivity? So using fMRI data and parcellation, we calculated the functional connectivity patterns. And using the functional connectivity patterns as feature, we trained machine learning models that predict pain ratings. In this step, we used multiple modeling options to generate a large number of candidate models. And then using a predefined set of criteria, including sensitivity, specificity, and generalizability, we selected the final model using the validation set, that is study 2. And with the final model, we conducted additional independent tests on multiple data sets. So these were the modeling options, including different parcellations and correlation methods and algorithms and hyperparameters. So in total, more than 4,000 models were created and tested. So we used these 7 predefined set of criteria for this model competition, including sensitivity, specificity across the training and validation dataset. And the winner was the model that used brain atom parcellation and dynamic conditional correlation and the principal component regression with 21 principal components. And this is the circus representation of the connectivity model, where the red lines indicate the strength of the functional connectivity between these two regions is positively predictive of pain ratings, and the blue lines were a negative predictor of pain ratings. The outer circle colors indicate the largest scale functional network membership, as shown here. Here I'm showing model performance in study 3 on independent test dataset. And this is the sensitivity test results. The mean correlation was 0.51, and we also tested the prediction only in the capsaicin run, given that the study 3 had a long scan duration that was around 20 minutes. And it also showed significant performance. And in this plot on the right, the black line showed the actual rating, and the red line shows the predicted rating. So they show quite similar patterns. And these plots show the specificity test results in the independent test dataset. And our tonic pain model was able to classify the capsaicin condition from the bitter taste and also aversive odor conditions with high accuracy. Therefore, we can say our tonic pain marker showed good sensitivity and specificity in independent testing. In addition, these are the results from supplementary data. And in this independent study, we delivered the liquid capsaicin for tonic pain and quinine for bitter taste twice within a run using MR compatible gustometer. And our tonic pain signature was able to track the tonic pain ratings with a significant correlation and showed 2 peaks at the time of maximum pain. In addition, the tops didn't respond to the bitter taste again, demonstrating its specificity. And the second research question was whether the tonic pain signature generalizes to clinical pain. So we tested our model on multiple clinical pain data sets. More specifically, the data were from chronic back pain patients, and we used the open data from openpain.org. And we found that our model was able to predict individual differences in pain severity of clinical back pain patients depending on tasks. For subacute back pain patients whose back pain were less than 3 months, the TOPS was able to predict their pain severity only when they paid attention to their spontaneous pain. In contrast, for chronic back pain patients, the TOPS was able to predict their pain severity even during the rest. In addition, the TOPS was able to classify chronic back pain patients from healthy controls across 2 datasets from Japan and the UK. Therefore, the TOPS seem to work for clinical pain, at least for low back pain. Lastly, we asked this question: how similar or distinct are the functional connectivity models among TOPS, clinical pain, and experimental phasic pain models? So before we compare those models, we examine the tops in more depth. So to help interpretation, we combined smaller parcels into more interpretable anatomical regions and then visualized the top positive and negative connections. So as you can see, the most important positive connections were found between frontoparietal and somatomotor networks, especially parietal parts of the networks. And for the negative ones, the limbic system and frontoparietal networks were important. So note that the brain regions other than the somatomotor regions appear to play an important role in tonic pain, and also the inter-network connections were more important for pain prediction, supporting the idea of distributed pain processing systems in the brain. So when we compared the tonic pain signature with other functional connectivity models, we found the significant network level similarity between the TOPS and the subacute back pain model, whereas the experimental phasic pain model was distinct from the other two models. And this plot shows the bootstrap-based network level comparisons between the model weights, and the results show that the 7 out of 9 networks showed shorter distance between the TOPS and the subacute back pain model compared to the experimental phasic pain model. So the TOPS is more similar to the subacute back pain model than the experimental phasic pain model. So to sum, we developed an fMRI dynamic connectivity based marker for tonic pain, and it showed high sensitivity and specificity in predicting and classifying the tonic pain condition. In addition, the marker worked reasonably well for explaining individual differences in clinical pain severity and thus provided a potential candidate marker for clinical translation. And lastly, tonic and clinical pain showed similar network level connectivity representations, which were quite distinct from experimental phasic pain. So I want to go over a couple of more slides for discussion of promise and challenges of developing neuroimaging biomarkers of pain. So one of the main reasons that our approach worked is, in my opinion, that we used the component process approach. So in our recent review paper, I contrasted this component process approach with this direct prediction approach. So in the direct prediction approach, researchers usually use the imaging data to directly predict the clinical outcome, such as pain severity. But in the component process approach, we aim to first develop brain markers for component processes, such as sustained pain, in our case, and then use them to predict the clinical or functional outcomes. And given that the clinical or functional outcomes are results of complex interactions among multiple underlying causes, this type of hierarchical framework might work better. In particular, in our study, we put more weights on within individual modeling to model a component process and then move to the between individual modeling. But the direct prediction approach often fails, maybe because they directly target the between individual variance, which is much more difficult to capture. So this is actually understandable because we now know that the pain involves distributed brain systems and have the degenerate neural mechanisms. So in other words, there are multiple routes for pain processing, multiple pathways, and each individual is likely to process pain differently. And even within an individual, pain processing in the brain could be different each time and each moment. So recently, we analyzed multiple datasets and trained one model per person and obtained a list of brain regions that are important for pain processing regardless of the weight consistency across individuals. And then, when we examine the pattern level individual variability of these regional pain representations, some prefrontal regions such as ventral medial prefrontal cortex and lateral prefrontal cortex regions show the highest individual variability, while the sensory motor areas show the lowest individual variability. So interestingly, the ranks of these pattern level individual variability was correlated with principal gradient from this recent paper by Margulis, and the correlation was almost zero point 0.7, suggesting that the individual variability in pain representations is closely related to functional brain organization, particularly the functional hierarchy. And therefore, in future studies, we really need to take these functional brain organization principles into account to develop better performing brain biomarkers of pain. And I want to end this talk with sharing an idea, actually from one of my graduate students, Jae-jung. One day, he came to me and shared his idea that future neuroimaging studies should address the 3C. The first C is comprehensiveness, meaning comprehensive mapping between high-dimensional pain experience, that is, pain types or qualitatively distinct experiences and different symptoms, and with multivariate brain representations. So currently, we are modeling one thing at a time, but to get a better picture of mind-brain relationship, we really need to pursue the comprehensive sampling of the high-dimensional space. The second C is causality. Given that the most of the current existing neuroimaging biomarkers are based on correlations, and thus future studies need to establish the causal mapping. So the causal mapping could be the mapping between brain representations and pain experience or behaviors, or there could be causal mapping between brain regions for pain processing. And third, clinical utility. So no existing studies convincingly demonstrated the clinical utility, and thus future studies need to aim to demonstrate the clinical utility even for one patient. And for these, we could use the dense sampling approach, or naturalistic neuroimaging, or computational modeling, and cross-species studies. And these are the research direction of our lab, and I hope you can be with us in this journey. With that, I want to thank funding sources and also our Choong-Wan lab. And here, Jae-jung and Lada did the most of the work that I presented today. Thank you for your attention.